Hello friends, welcome to my channel Online Classes Dr. Anand Pandey. Today we will be dealing with the subject of plant biotechnology, which is also in the main curriculum of BSc and MSc. So in this class we will be understanding about various aspects of genetic markers, how this concept came into being, and what are the various characters we should know for understanding genetic markers. Before going into detail about genetic marker, we should understand what is variation. Actually, variation is nothing but difference in the characters, difference in the different individuals, difference in the various set of morphological, anatomical, cytological, and biochemical characters existing between two different plants. And how these characters come into existence, it is because of the genes. These genes are made up of DNA, and DNA is packaged in a chromosome pair. The genes actually are responsible for providing the various inheritable characters to a plant, and as such, they provide these visible traits which we called as phenotype, and the important gene which is responsible for creating that trait is called its genotype. In this way, genes provide different sets of character to the plant, and these genes are located on some specific segment of each chromosome. Now, what is marker? Here you can see a pointer can be seen, which is easily visible in the Google Maps. Now, why I have shown this pointer? Because when you use a Google map, you easily get the desired location of your place of interest when you search in the search box of the Google map, and this pointer shows the exact location on that Google map. Likewise, a genetic marker is also a similar thing which tells about the location of some gene of interest, because when you understand a gene library and when you search a gene, it is the gene of interest which you search in a gene map, and in that gene map or in that genotype, it is the marker which enables us to let us know the location of that gene, and as such we identify the desired gene. Thus, now we must know what is gene and how a gene is different from a marker. Actually, genes are responsible for the production of some desired proteins in a plant, which provides some desired character or undesired character also, depending upon the protein type. Means how the protein is reacting in the metabolism of the plant. It is that metabolism, or it is that reaction which makes it beneficial or harmful. And when we have to identify a beneficial protein or a beneficial gene of interest, we simply identify that gene of interest by the presence of a specific marker sequence, which is a specific DNA code, genetic codes, which are present either before or after or in between of that gene of interest, and the markers are linked to the gene of interest. Now, this another important term, linked, means it is linkage. We have to go into detail about linkage, but I will tell you in short that linkage is the ability of two genes to move together. Okay. Likewise, a marker, when it is linked to a gene, it moves together with the gene when it is transferred from one uh, generation to another generation. So when we search for any gene of interest, markers help in enabling us to identify that gene of interest. Now, keeping all these facts into mind, Genetic markers can be defined as genetic markers are the biological features that are determined by allelic forms of genes or genetic loci and can be transmitted from one generation to another, and thus they can be used as experimental probes or tags to keep track of an individual, a tissue, a cell, a nucleus, or a chromosome or a gene. There are some other characters of the genetic marker also, like they do not represent the target genes themselves but acts as a sign or flag. You can simply understand it by a landmark. When you move into a new place, you simply search for some landmarks. Like if you want to tell that I want to go some place near Subhash Charaha, then that Subhash Charaha is the landmark. Or you want to tell us that, okay, you meet me near this shop, or you meet me near this, that landmark is the genetic marker. So it is the closest thing to that gene and tightly linked. So a genetic marker is always present in a very close proximity to a gene. Means some marker which is present on a gene locus which is away from the gene of interest cannot be taken as a genetic marker used to identify that gene because it is very away. So you have to take a long stretch of that marker sequence to used as a primer. So it cannot be taken as a marker. Means a marker should be very in a close proximity of a gene. So classification of genetic markers: classical markers, morphological markers, cytological markers, and biochemical markers. So morphological, cytological, and biochemical markers, these all are the markers which can be easily viewed by some basic test. Like morphological markers can be easily viewed as tall, dwarf, or various genetic traits which you have read in Mendelian genetics. These all are morphological markers. Cytological markers like presence of B chromosomes, biochemical markers like production of some specific chemicals like vincristine, vinblastine in Catheranthes roseus, then presence of thymolin, TMI, trichosamami, and presence of opium, morphine, caffeine, and likewise these substances which are secondary metabolites and act as a marker also, biochemical marker. And likewise DNA markers are various DNA sequence or other sequences which can be either artificially produced or they are present naturally and act as a specific marker in identification of some gene of interest. I am always pressing on one point, that gene of interest. Only markers are produced for those genes which are some beneficial to the biotechnology or they help in some getting some desired character. See, these are the various examples of morphological markers, basically phenotypic markers, and these are naked eye markers: food size, food shape, food color. Cytological markers and protein markers. Likewise, various other markers can be discussed. So, what is a molecular marker, which we are going to deal in great detail? So, molecular markers are associated to a gene and are naturally occurring DNA sequences that are very close to the specific gene sequence. See, here you can see that how the um, organization of the DNA, organization of the genes is taking place in the uh, plant cell, that inside a nucleus there are various chromosomes. These chromosomes are highly condensed in nature and have various exonic and intronic sequences. In those exonic sequences there are various genes of interest, and suppose this is some gene and this is some marker for that same gene. So that marker gene will be used for the identification of the particular gene. So thank you for patience hearing, and you can easily comment your queries in the comment box, and we'll meet you in the next lecture. Thank you all.